Thank you, everybody, for joining for this week's expert talk. The presentation tonight is Chloroviruses in ALS, and our expert presenter is Dr. Gary L. Pattee, and we're really happy to have him here. Some housekeeping really quickly. Of course, every program like this in our expert talk series is both a presentation on updates in research and other information that comes from us from world-renowned experts. It's also a time for you to be able to ask your questions directly. So while Dr. Pattee is getting ready to present, we will also remind you that you can ask your questions in the chat. We'll try to get to every single question during the Q&A period at the end of the presentation. And of course, as always, we welcome everybody to be a part of our ongoing observational studies in speech and our other studies that you can learn more about at everythingals.org/research. And of course, we're looking forward to a busy year of trial updates, of educational programs, and some new programs coming as well with the likes of great known experts who are always here with us, Dr. Lyle Ostrow, who is here, as well as Richard Bedlack and others who will be joining us throughout the course of the year. So we're excited to kick everything else off this year, and I'm going to introduce Dr. Pattee now. Dr. Gary Pattee is a physician scientist specializing in neurology and neuromuscular medicine in Nebraska, with a career spanning more than 3 decades. He's earned his medical degree from the University of Texas Health Sciences Center at Houston in 1986 and completed an internal medicine residency at Creighton University School of Medicine in 1987. Dr. Pattee followed this with a neurology residency in 1990 and then a neuromuscular fellowship at the University of Colorado School of Medicine in 1991. In his dual role as a clinician and researcher, he has dedicated his career to advancing understanding and treatment of neuromuscular. His research has addressed biomarker development, clinical assessment of bulbar dysfunction, and ALS therapeutic trials. As chair of the NEALS Bulbar Committee, he leads efforts to refine evaluation and management of speech and swallowing impairment in ALS. With that said, we are so thrilled to welcome Dr. Pattee to present. And Dr. Pattee, the floor is yours.
Well, Mr. Rubio, thank you so much for that very kind introduction. And thank you for the opportunity to share some of the work that we've been doing with everyone who's chosen to tune in. As mentioned, we're talking about a virus tonight, something called a chlorovirus. I'll go into a bit more detail as we go through the talk, but it's just a recently identified virus that we feel may be playing a role in neurodegenerative diseases. For starters, and I wasn't clear to the audience, I thought we'd just run through some quick overview in terms of ALS. When we talk about ALS, we're talking about three words, in essence. A, myotrophism. A, lack of myomuscle trophism growth. The lateral sclerosis has to do with the corticospinal tracts within the spinal canal. So we get what we determine, or what we describe as an upper and lower motor neuron pattern of presentation. There are few diseases that really show this type of a picture. When we're talking about ALS, we're talking about terms: fasciculations, involuntary muscle contractions, activation of individual motor units, spasticity, increased muscle tone, exaggerated tendon reflexes, and then motor neuron degeneration, which affects both the upper and lower motor neuron, in essence, all of the corticospinal nerves. It's classified typically as sporadic or familial. The sporadic case is the majority of what we see, anywhere from 90+%. In other words, we don't have a known cause for a sporadic case. Familial can be up to 5 to 10%, and there's multiple genes that have now been recognized. Environmental has to do with what we describe as the toxic exposures, sometimes referred to as epigenetics, and they can play a role with other genetic susceptibility genes, that all of which can potentially turn on a motor neuron process. The wide spectrum of pathologic findings, which I'll talk about in a bit more detail, suggesting a syndrome rather than a single disease entity, which has been part of the problem when it's I think when trial designs for drug trials have been put together, we have to keep in mind this is a widespread disorder, not just a one process we're going after. motor neuron atrophy, motor neuron loss are what we typically see with this condition. And so this is just a bit of an anatomic picture showing the widespread nature from the cortex, the motor cortex, into the cortical spinal fibers of the spinal cord, and the bulbar motor nuclei into the bulbar nuclei, and then on into the anterior horn cells and involving the muscles. So we've got a disease process that is quite widespread in nature. Risk factors have been identified with ALS. Some of those are the genetic. We'll talk about something called the SOD1 gene mutation, TDP43, C9orf mutations, other TBK, other mutations have been identified, all of which pose risk factors for this disease. Environmental factors have been a focus, a hot focus in essence, looking at heavy metals and a variety of different pesticides, electromagnetic field exposures, nutritional state, all of which can play a role with this disorder. None of which, at least with the environmental factors, are a one-to-one connection, but they all seemingly have played some role. Now, focusing on viruses. Now, infectious agents such as viruses have been postulated as contributors to motor neuron disease. And the list at the bottom include those that we've identified. West Nile, poliovirus, of course, we all are quite familiar with. Thankfully, we're not seeing much of that any longer. HIV, cyanobacteria is not a virus, it's a bacteria, but it is an infectious agent that can be tied in with motor neuron disease. And then the human retrovirus K, HERV-K, Avi Nath has really pushed that virus forward in terms of being part of our virome, playing a role with motor neuron disease. So we do have data suggesting viral involvement. This is just a quick list, not to go into detail, of articles in the literature just describing viral infections and inflammatory mechanisms playing a role with ALS. Rick Bedlack had a publication within the last few years regarding a COVID-19 accelerated disease progression. And these were two patients with slowly progressive ALS who experienced a rapid decline after developing SARS infection. So it really implicates, if the virus isn't necessarily causing, it can certainly contribute to the rapid progression of an underlying disorder. Chloroviruses, just to discuss this in a bit more detail, represent an environmental exposure only recently associated with certain human pathogenic conditions. Now, these are described as large, double-stranded DNA viruses. They're located worldwide. They were first identified in Germany. They're located in freshwater environments. They infect and replicate in what are called chlorella-like green algae. So the focus that we've at least have been zeroing in on with our lab are the green algal blooms and the potential health threats that might pose. The CV variants have been shown to infect mammalian macrophages. Now, the macrophage is the organelle that oftentimes carries inflammatory or anti-inflammatory mechanisms. In this case, the macrophage triggers inflammatory mechanisms we call cytokines, and we saw this with infections in mice, also inducing neurocognitive deficits. So we do have some pretty good data identifying that these viruses basically initiate an inflammatory mechanism. The one that we've been focusing on, the type of chlorovirus, something called ATCV, it's a member of one of 4 known chloroviral types. What got our interest is that it encodes its own functional, something called a copper zinc superoxide dismutase, the SOD1. So SOD1 is one of the first mutations identified as to a cause of ALS. So this particular virus encodes its own SOD1. Now, the second point here, it's predicted that the SOD1 lowers the concentration of something called reactive oxygen species in cells early during a viral infection, evading thereby the innate antiviral resistance. Basically, it helps protect the virus from being attacked by the host cell and allows it to replicate and perform its duty to survive. Now, this is of clinical importance, as ALS variants are due to polymorphisms in the human SOD1 gene. And that's been quite well identified, and we've done a lot of work with mouse models of SOD1 gene. So the importance of abnormal SOD1 in ALS is also represented in the sporadic cases, accounting for most cases, as I mentioned, where evidence of SOD1 dysfunction has also been identified, leading us to conclude that the influence of an ATCV1 SOD1 in the pathogenesis of ALS may be significant. So the hypothesized pathogenic effect of ATCV1 on humans, as I mentioned, a recent discovery. It was thought to only infect certain eukaryotic chlorella-like green algae, such as something called Acanthocystis trophaceae, a common protus of inland waters. Yet infectious chloroviruses are found throughout the world and at times show major spikes. We saw a fair amount of it in late summer, and we see a fair amount in the spring, where we see green algal blooms in fresh waterways. 43 chloroviral isolates have been sequenced, classified into 4 basic groups. Not all aquatic systems, however, contain these viruses from each group, yet the chance of human exposure appears to be quite high. And as mentioned at the bottom, it now appears that exposure to these is highly significant following a recent detection of chloroviral DNA sequences in the human virome, in the oral pharynx, identifying it as something that most likely most of us have been exposed to. So the hypothesis is, in essence, a corollary of a well-established theory that high levels of neuroinflammation not only impair cognition and behavior, but also neurological function. Thus, we're evaluating this hypothesis that exposure to or infection with this particular virus may lead to a motor neuron disorder. What we started by a few years back is just looking at blood from our ALS patients. And we looked for something called an anti-ATCV1 IgG subclass, in essence, just the IgG inflammatory antibodies within an individual's system directed against the virus. And what we identified was that patients with ALS had a much higher titer than the healthy controls. Of note, they all had some titers, some prior exposures, but the ALS patients in the blood had much, much higher and statistically higher than what we saw in our healthy controls. This was mainly with what's called the IgG1 subclass of IgG, which was higher in the ALS patients. And it's the most abundant IgG subclass because it mediates response against viruses, which caught our interest. Therefore, the summary point, ALS patients would therefore appear to have had a significantly greater exposure to ATCV1 or a greater antibody response to a similar level of exposure compared with healthy controls. So once we found this, we talked to some colleagues and suggested that we try to put this in an animal model. We'll get to that in a second. This shows the data from the serum study in sort of a graph form that the ALS patients had a much higher and a statistically significant elevation of the IgG1 anti-ATCV1 in their blood compared to the controls. We went ahead and obtained some animal studies, and we used a transgenic SOD1 mouse model, injecting animals with the virus, and had some of the transgenics without the virus, and just see how they manifested. And as the top point suggests, the infected mouse macrophages were looked at, and they supported low levels of the ATCV1 replication in vitro. So after we infect these animals, we looked at the macrophages, and lo and behold, they were churning out inflammatory mechanisms in those infected animals, suggesting that persistence of this virus within the macrophage may stimulate production of immune factors. So we found that by injecting the animals with the virus at an early stage led to a progression of their motor weakness. And then dissecting it out a bit, we found that within the macrophage, this virus existed and produced an inflammatory cascade, suggesting almost like a Trojan horse phenomena. The virus can exist within the macrophage and be transported throughout the body to different cellular systems and continue an inflammatory infectious process, leading us to hypothesize that ATCV1 persistence in macrophages may induce inflammatory cytokine production, and that could be a mechanism by which neurological dysfunction occurs. So this graph it shows the life cycle of these SOD1 animals, and they typically live to be maybe 155, 160 days. What this is showing, however, is the motor decline. And in black and red, we have two forms of virus, the ATCV and the PBCV. And as you can kind of see from the graph, at about day 140, these animals start going downhill. The mean clinical score, it's a 4-point score showing motor weakness, 4 being the worst, actually. But the animals that had been treated with the virus went downhill much quicker than the saline-controlled animals, yet that didn't affect survival, which we found to be of some interest, certainly. It did, however, trigger a faster propagation. We also looked at what we call wild-type mice, which are mice that didn't have any evidence of a transgenic genetic disorder. We gave them the ATCV1, and over a 2-year life cycle, they showed no evidence of disease, suggesting that it may take a genetic mutation of sort that this virus may trigger to turn on an ALS deterioration or an ALS motor neuron condition. To summarize, ATCV1 SOD1 augments production of inflammatory factors, interleukin-6, nitric oxide, ISG expression, all associated with the development of ALS in humans and motor neuron disease in our transgenic mouse model. Unanswered questions, however, the mechanisms through which humans are exposed, remain largely unknown. Respiratory inoculation, oral ingestion may be a means of significant exposure. Given that measurable serum antibody titers against this virus have been detected, a common means of exposure worldwide appears to be evident. There is transit through the blood-brain barrier, and we'll get to that in a second, and that may be a means of how the virus induces immune mechanisms targeting the central nervous system. We ran another recent study, which we found very interesting. We requested and received NEALS biorepository samples. So the NEALS is the former Northeast ALS Research Consortia, and they have a biorepository of blood and spinal fluid from ALS patients throughout the country. They sent us 37 of these samples, both blood and spinal fluid, and we analyzed these samples for the IgG ATCV1 antibodies. And of note, we did find elevated levels of these antibodies, both ALS serum and spinal fluid, suggestive of a chronic immune exposure. This was something that we hadn't identified or been shown before, and it raises some concerns. The mechanism of human exposure is still unclear. Direct, indirect, oral, respiratory exposure appear most likely. We're currently looking at therapeutic targets, potentially antiviral agents, which might have an effect on this particular virus in animal models. Now, here's the data that was really quite interesting to us. The serum on the left, the ALS patients in blue, healthy control in red. But of note, the spinal fluid in the right side, once again, ALS in the blue, significant p-value was achieved. This really was, I think, what was most interesting to us and to a lot of people we've shown this to. How is it that you're getting an IgG antibody response to this virus at a much higher rate in ALS patients over healthy controls? And what does that mean? And this has raised even more questions than answers. But if we spend a little time looking at some of the neuropathology behind ALS and chlorovirus, and I don't mean to get too detailed, but this I felt was important to kind of review. Off to the left, we see mechanisms, not all, but many of the mechanisms thought to be involved in ALS disease progression. And yet, we may also see something very similar in what the chlorovirus can enact. And we'll go through each of these. Oxidative stress. Virus-induced mitochondrial reactive oxygen species, in parentheses, improves viral replication via modulation of host pathways. In other words, the virus likes to take over the cell, and it does have an effect on the mitochondria, and it does trigger an oxidative stress, which is sometimes referred to as reactive oxygen species. Viruses interfere with the mitochondrial electron transport chain. Now, early data has shown that electron transport chain complexes 1, 3, and 4 are abnormal in human ALS, and this virus seems to have an effect on that mechanism. Replication within something called the endoplasmic reticulum, typically responsible for protein production, does lead to increased stress and more production of the reactive oxygen species. Also, viruses deplete antioxidants, which work to control the reactive oxygen species, such as glutathione. So oxidative stress may in fact be playing a role with this viral infection. Cellular transport, how the cell transports information or nutrients throughout the cell. We know that the chlorovirus, once it infects a cell, depolarizes the plasma membrane upon infection, inactivating these secondary active transporters. They can rewire the host transport system, so they have their own efficient replication factory, but cellular transport does suffer. something called nuclear pore dysfunction, a hot topic of investigation right now. In essence, that's the gateway between the nucleus and the cytoplasm. That's how information is transited from one area to another. And what are called RNA binding proteins are mislocalized now to the cytoplasm. TDP43, for example, triggering aggregation. So there's been a lot of research on is the nuclear pore potentially one of the first upstream mechanisms when ALS is occurring. And what we see that the chloroviruses target the nuclear pore complex through viral protein binding and cleaving of certain nuclear pores, resulting in structural changes and impairing the normal gating function. Now, this disrupts the normal nuclear trafficking, as I mentioned. It blocks essential host mRNA and protein transport, controlling entry and exit transport of host gene expression. It overcomes the nuclear pore's gateway role. In essence, it hijacks the cellular machinery. And this could be a key factor in terms of propagation of disease process. Now, axonal transport, that is one of them, that's like the highway of the cell. The chlorovirus hijacked the host cell transport, as I mentioned, leading to depolarization favoring replication. It also facilitates viral genetic material delivery to the nucleus. Viral exploitation of the host cell axonal transport for purpose of survival replication take place. So another means of the viral involvement of a critical mechanism of cellular function, one that is implicated in ALS. As we talked about earlier, mitochondrial dysfunction, these viruses induce mitochondrial stress, disrupt the processes, including the nutrient transport, leading to metabolic shifts, reducing what's called the ATP, increasing the reactive oxygen species within the cell. They induce cellular stress, ultimately leading to dysfunction, disease propagation, apoptosis, and cell death. Mitochondrial dysfunction, a key part with ALS, and also involved with this viral infection. Neuroinflammation, we've touched on before. Our mouse studies showed that elevated levels of inflammatory mechanisms, the IL-6, INOS, inducible nitric oxide synthase, interferon gamma, all pro-inflammatory markers have been shown to be triggered through this virus infection. And within the human oral pharynx, we found DNA sequences that have been associated with cognitive abnormalities and visual processing motor speed. This was done several years ago at Hopkins. Microglial activations, cytokine release have also been identified in animal models, all playing a role in the development of neuroinflammation, all of which we know is involved with ALS. RNA metabolism. Chloroviruses reprogram host nucleotide synthesis pathways. They encode, as mentioned, their own transfer RNAs, also interfering with the host RNA, just to allow the viral survival. These viruses, in fact, they infect these cells rapidly, and they degrade the host cellular machinery to take control for their own transcription and subsequent replication. So it's a survival mechanism, and it's fairly amazing how many mechanisms these viruses basically engage in for their own survival. And a mechanism of excitotoxicity. Similar to the indirect effect of viral-induced neuroinflammation, we do see excitotoxicity. The inflammatory factors likely disrupt neuronal function, affecting neurotransmission. And chloroviruses, we've already identified, induce inflammation, which have secondary effects on the excitotoxic mechanisms, glutamatergic mechanisms, for example. And then, just to kind of summarize the routes of transmission, we simply don't know the answer. We're concerned about freshwater exposures, airborne transmissions, increased risk during algal blooming seasons, chlorella-based nutritional supplements. Interesting how many of these supplements are now out. We've also wondered, when opening the packaging of these powder supplements, if the respiratory exposure to the powder as the box is opened might be playing a role. We know that the virus resides within the microbiome of the gut in the oral pharynx. And we've had concerns now with the green algal blooms in freshwater ponds that farm-raised fish products, crustaceans, shrimp products, might be exposed or feeding on these green algae with viral infection. And could that be a means of potential propagation of the virus into humans? None of this has been proven. These are our concerns. So this last slide kind of summarizes much of what we were just talking about. We have the cell body, we have the axon, the dendritic projections, we have the microglia or macrophage, the innate immune mechanisms, we have the supporting network of astrocytes, and we look at this ALS sort of model. And we see that we've identified, at least with ALS research, impaired glutamate intake, impaired excitotoxicity from glutamate. As we mentioned, SOD1 aggregation, mutations in various genes, mitochondrial dysfunction, neurofilament accumulation, and transport of cellular dysfunction taking place, release of inflammatory mediators. What can do all of these multiple mechanisms apart from something that has multiple sort of fingers to it, such as a virus, as I just showed you? Now, we've only really studied this particular virus. We haven't looked at others, but it's been an interesting series of experiments over the last several years. And every time we do a study, we step back and say, now, what do we do with these results? Where does that take us next? But it leads us to several conclusions. And I'll run through these basically and hopefully open up for questions. Exposure to the ATCV1 may accelerate. It's all may. We don't have definitive answers here. The onset of an ALS-like motor neuron disease, certainly in the transgenic mouse model. But these chloroviral SOD1 also augments induction of inflammatory factors from macrophages. Point 3, chloroviruses may trigger many of the pathologic processes implicated in ALS, as we just reviewed. An elevated ALS patient spinal fluid chloroviral titers are strongly suggestive of CNS involvement. In essence, our cumulative research findings would indicate that SOD1 encoding chloroviruses, and not all have the SOD1, the ATCV1 does, that's the one we've been studying, which are ubiquitous within freshwater environments worldwide, appear to be associated with the pathogenesis of this disease. A few years back, we did have a publication on some of our early work. If anybody's interested, it's in the Frontiers of Neurology. You could go back and look at it. I think the key point I want to make is, until we learn more, avoid the green waters, okay? This is exactly what we don't want people to be exposed to or nearby. And this is taken from a stream not far from my home, believe it or not. And this is representative of the green algal blooms that we see in the spring and in the summertime. Not to be confused with the blue-green algae that we see with the cyanobacteria, the BMAA mechanisms, which are a bacterial infection. Yet, the blue-green and green algal can grow together, so we may have more than one toxin that plays a role. But I tell people to stay away from these waters. Keep your animals, your children, out of this green water when you do see the green algal blooms. That, in essence, is summarizing much of what I want to discuss. I'm certainly willing to talk any questions that you might have, if you have any on some of the slides. I had the slides numbered. That might sometimes be helpful, but open for any discussion at this time. Thank you for your attention.
Thank you so much, Dr. Pattee. That was a great introduction and then coverage of not only what you're studying, but the potential causes for it that I think did trigger a lot of good questions. So we'll jump into the questions here. Anusha is going to join me for we're going to take turns kind of reading them out to you from the audience.
Sure.
The first one is from an audience member who said, I would expect the COVID cases either involved comorbidities with zinc deficiencies or that the COVID itself caused a zinc diversion attempt to block the virus. Is that something that's been part of your studies or has been linked to anything?
You know, that's something that is being looked at, not part of our studies. We haven't looked at the COVID virus itself. There has not been, to the best of my knowledge, an overabundance of COVID-associated ALS cases. I will bring up a side note here. I did have a case that I'll share briefly of an individual who had a COVID infection in July one year. I saw him in March the following year. Rarely when I see a patient for the first time can I diagnose ALS, but unfortunately this was the case. And I asked this young man his story, and he basically said, well, there was some family history. And as it turns out, his mother and his uncle both had ALS. And we ended up doing gene analysis, and he had a tank-binding kinase mutation. And what was interesting about this whole story is that his mother and uncle were in their 60s when this disease became manifested. This young man was in his 40s. So could the COVID infection have basically triggered the onset of this disease? And that's one of our assumptions, that it potentially could be viruses playing a role with disease mechanisms. Another quick example, we know that Epstein-Barr virus, the cause of something called infectious mononucleosis, is now somewhat highly associated with MS, such that many people have antibodies to the EBV in their system without any symptoms of neurologic impairment. Yet the majority of patients with MS have positive EBV titers, suggesting that this particular virus may, either lying dormant or having an effect earlier in the life cycle, be playing a role in the development of MS. And that's been fairly well established, actually. We've seen COVID cases where two individuals, a husband and wife, can both come down with COVID. One gets the sniffles, the other one's on a ventilator. And it gets back to how does that virus affect two people differently? It has to do with so many other factors, mainly their immune profile to begin with. So there's a long answer about COVID, for example. RNA virus, a bit different than the DNA we're studying, that we certainly don't understand. But a lot is being looked into the whole concept of will COVID play a role downstream. It may be too early to know with motor neuron disease.
Thank you.
Got it. Thank you so much. All right. The next question is, how would you explain that in spite of high levels of inflammation by the level of IgG, that no anti-inflammatory drug trials were successful?
So this is an excellent question, and this is the one we all ask all the time we get together. And that's why I think the last slide showing the multiple pathogenic mechanisms at play with this disease quite likely are helping to explain. In other words, everybody may be a bit different, you see. It could be that what will be an anti-inflammatory mechanism in one individual may not be the same for others. And what we really are lacking are biomarkers showing the putative mechanisms of disease propagation in each individual patient. And if it's more excitotoxic, if it's more mitochondrial, if it's more inflammatory, we could aim, if we have these drugs, basically a cocktail of medications aimed at trying to slow it down. I think the best example is, if you've got a scenario where, and I use this example for my patients routinely, if a house is burning and I can successfully put the fire out in the dining room and maybe the den, yet the kitchen and master bedroom are still ablaze, I'm going to lose the structure. So if I can't address all the mechanisms that are affecting that particular tragedy, so to speak, they're going to continue, and that's going to ultimately be the main issue we see. We've been fairly fortunate to see that drugs such as riluzole and Radicava has had some positive data suggesting there has been some impact, but these have not been profound. I think what's been most profound have been the upstream effects of the medications that we've been, the Qalsody medicine, the SOD1 agents that we've been finding for our genetic patients. I'd love to see those studies be performed in sporadic cases, because some of us believe that the SOD1 is dysfunctional even in the sporadic cases, and potentially this could have a benefit. Catching things upstream is really the answer. And I think seeing patients as early as possible and hitting them with everything we have. But I don't believe one mechanism, if that's what we're attacking, will have a significant impact on the overall disease progression.
Thank you so much. All right, I'm going to go to the next question, which you started to answer partially, so I'll ask it again, and we can. But there's a second part to it that's a little fresher that we'll delve into. Since exposure to chloroviruses seems to be pervasive, do you think certain people have a genetic predisposition, which I think we talked about a little bit about, to an increased antibody response to chloroviruses, which then might worsen or cause? Alternatively, maybe people living with ALS just have broadly enhanced inflammatory or immune reactivity, and the increased chlorovirus antibodies are not specific. You can speak to both of those, or if you think there's kind of a relationship between the two, by all means. So.
No, excellent questions. The last point, we have done some other looking at tetanus toxoid in individuals, looking at other immune mechanisms, and we're not seeing an across-the-board drop in immune activation or immune mechanisms in our ALS patients, suggesting that we don't think it's across the board. They just don't have the immune tolerance to deal with this particular virus. In terms of, is there a genetic predisposition towards suffering from viral infections? Excellent question. I think we'd have to pose that with a lot of other viral disorders that we see. We haven't identified anything. I'm not aware of any genetic disorder that would lead to a higher predilection towards a viral infection. but there could potentially be some immune mechanisms at play. There's obviously a lot about the immune system, viruses in particular, that we don't quite understand. But I've always been somewhat curious. Is this something that we're going to be, as we look at more viruses, we're going to find more and more foundational principles? It's also important to identify if, in fact, and it's a big if, that this virus is playing a role with this particular disease. How do we identify it early on? Just as another aside, we have looked at communities where they have had fresh body waters which have the virus within it. And of note, we found a higher incidence of disease in communities or areas that are on the east or southeast edge of these waterways. Case in point, there's a town here in Nebraska that has about 23,000 people. It has a very large lake in the middle of the town. We happen to have 3 patients with ALS from that town, and they all live within a few blocks of each other on the southeast corner of that lake. Now, with 23,000 inhabitants, knowing this is 1 to 2 per 100,000 incidents, this is something that catches our attention, making us wonder, could it be that something such as a chlorovirus, as it sort of winds blow from west to east here in North America, could that be playing a role in transmission of potential disease? A lot of big ifs there, but you can't ignore these associations. And what might they be telling us?
Thank you
Thank you.
so much. All right, so the next question is, was the membrane depolarization the mitochondrial membrane or the cell membrane?
So good question. It's the cell membrane. Ultimately, however, that will lead to calcium entry, and with something called the mitochondrial permeability membrane also becomes affected with ALS and with destructive processes. So initially, what that virus does is it depolarizes the cell membrane, changing multiple mechanisms within the cell, favoring the harboring of the virus, its transcription, subsequent replication.
All right, thank you.
Thank you. What biomarker should we look for in patients if chlorovirus is involved, and what is the most practical next step toward clinical impact?
Yeah, excellent question. The cheap answer is we're not there yet. I guess we don't have a commercialized chloroviral IgG titer available. This is all done within our lab. Yeah, I think it'll be important to look at more samples. I think it'd be important to establish what's an abnormal level of IgG1. We see elevated levels, but we haven't had a cut point necessarily that shows above this particular parameter we would consider this to be abnormal. Ideally, that would be a next step if, in fact, we have more data supporting this virus playing a role in the pathogenesis. But the next step is, well, then what do we do about it? There are some studies that are underway that are looking at something called lectin receptor inhibitors. Not to get too technical, but the macrophage, which we think harbors these inflammatory mechanisms, it must sort of team up with the virus. And if we can inhibit that viral capsid macrophage lectin receptor interaction through these lectin receptor inhibitors, agents that we're looking at now in our lab, that potentially could lead to a reduction in the propagation of disease. A lot of ifs, but that would be one mechanism that we could potentially interfere with. It's really hard to get rid of chlorovirus. They're everywhere. They're all throughout the world. Now, a lot of people have had exposures to them, as we see in the normal human controls. They all had some titers. What leads to a higher titer? What leads to a higher exposure is the perplexing question. And I guess we're still sort of stuck on, why do these ALS patients, in the majority of them, have these high ALS or these chloroviral titers within their spinal fluid? It suggests something there that we yet to have been identified, but something we hope to continue to work with.
Great, thank you. So this is kind of along those same lines, but do you check for ACTV1 antibodies in your own patients? And then how can people get tested for chlorovirus exposure, and what is the associated cost for patients as well?
These are excellent questions, and I guess just to temper it a bit, that's still not we're still not at that point yet. We're not to the point where we're saying we're sure that this is playing a role. We still have work to do to show if in fact it is playing a role. But point by point, no, I'm not testing HCV1 in my individual patients, because in essence, I really don't have anything that I would offer differently. And we don't have a commercial way of doing so. We have done it in a bulk sample from our labs, but ultimately that might be something as the studies progress that we would want to embark on, or get labs that might be interested in pursuing that. And then more importantly, at the same time, what can we do to impact that virus? So when we're talking about biomarkers, typically these are not necessarily tissue, but these are serologic markers. I mean, as you're, I'm sure, somewhat aware, neurofilament light chain is something we use routinely in our ALS studies. It would be really nice to see if we could find, maybe the IgG1 antibody for this particular chlorovirus as a marker, in essence, of its infectivity. And if elevated, if we had something we could go after it with, that would be the next step. And that is really what we're working on now.
Thank you. The next question I'll take is, well, it's a little bit of a rehash. If we asked about COVID, but are there other viruses, whether they're respiratory or others, that are prevalent that are being kind of get the same kind of scrutiny? I know early on we mentioned HERV-K as well as part of your bio. Are there other programs that you're involved in that kind of take the same methodology and that there are other viruses that are as tied to ALS onset or have the environmental factors that you're seeing, cellular, environmental, or otherwise?
We certainly have. This has drawn a lot of interest. Quick answer, our lab is really just working on the chlorovirus. But to expound on your question, as I mentioned, we see with all sorts of viruses that we've already identified. HIV, for example, we've identified patients with HIV infections who have developed an ALS-like phenotype. Interestingly, when their HIV is treated successfully with the combination of protease inhibitors, we see improvement of the ALS features, which is pretty amazing. We've obviously seen it with the polio virus that goes back a ways. That presents just with a lower motor neuron, but it does have an effect on the motor neuron itself. We had case reports of West Nile virus inducing an ALS-like syndrome in a multiplicity of cases. I've mentioned this cyanobacteria, which is something we're not studying. It has been studied extensively, also contributing to a motor neuron disorder. That's an infectious inflammatory mechanism. So I think there's a lot to be said about environmental exposures to infectious agents and their effect on human health. Of note, there was a trial done many, many years ago by some individuals looking to see, is the immune system playing a role in ALS? And to test that, they did what's called total lymphoid irradiation of ALS patients, meaning they radiated their entire inflammatory immune system and took out everything. And yet it didn't change the course of the ALS disease process. So they concluded that inflammation wasn't the mechanism. I don't really agree with that conclusion, but I think it also suggests other mechanisms are at play. We do know, I think, with pretty established data and literature, that there is a significant inflammatory cascade. Are viruses the key players? Could other mechanisms be playing a role? What factors within one's immune profile potentially make them more susceptible? These are all the questions we're trying to answer. And I honestly think it's just wise to be very careful of putative exposures. And if we're just going to selfishly focus on the chlorovirus, the slide I put up showing that I would avoid chlorella like nutritional supplements. I've seen these in health food stores. I shudder when I do. I just don't think we know enough that these are safe, and I'm not convinced they are. I think when you see the certain blooms in waterways, stay out of those waters. Keep your animals out of those waters. I think just certain, I won't say common sense, but certain basic principles to avoid exposure, I think, is the best approach we can take at this time.
The next question is really about we prevent chlorovirus exposure. And so I had some specific questions about environmental exposures. One was, could the airborne dust of animal feed be a source of infection? And the second was, could mouse droppings or a barn environment be a source of chlorovirus infections?
Yeah. Well, these are interesting questions. Of course, none of that has certainly been explored. The dust raises some real interesting concepts. We think back on the Gulf War syndrome and how there was a higher incidence of ALS reported in returning Gulf War veterans. There's some concern that maybe within the dust of this area, Kuwait and Iraq, that there may have been spores, there may have been other infectious organisms. Some have even postulated the BMAA may have been part of that dust, for example, that people could have been exposed to. In terms of animal droppings of these nature, I don't know if we could make a strong case for that. I got to admit, I've been a little leery to eat farm-raised fish and shrimp just because they're packed into small waterways that are. We've measured, interestingly, people from our team, high chloroviral titers in some of these farm-raised ponds, believe it or not, and there's no doubt that those animals within the ponds are exposed to the virus. And could that be a way of propagating a disease? Certainly possible. We just don't know. But I think it all has to be sort of tempered with some I think not a irrational approach, but a common sense approach. We've got to live our lives. We've got to be exposed. We're even in the water. We don't know if the drinking water has the potential for this viral infection. We know that we haven't identified anything quite like that, and treated water would probably inactivate most of these viruses. But this is a virus that was really started in terms of it being explored in existence in the 1980s. It's only been around for about 40 years. People have known about it. It's a very large virus. And as such, it's considered a giant virus. There's never been a lot of work looking at human disease. So we've been sort of embarking on that on our end, and we've been finding some pretty interesting results, raising some concerns. I just think that diseases like ALS span multiple mechanisms. It could be that in inflammatory exposure, viral exposure, as we've seen with other viruses, could for some people be a means of maybe propagation of a disease already destined to occur.
Thank you. I'll take the next question. How does it affect TDP-43, especially with sporadic cases it being 90% of reported ALS cases, and its pathology identified with misfolding of TDP-43?
No. And so it gets back to some of the work we did that showed people who have, in this case, I'm assuming they're a TDP-43 mutation, and the TDP-43, just as a review, is an RNA binding protein. It resides within the nucleus, but it's mislocalized to the cytoplasm, where it interacts with other cellular mechanisms, leading to protein aggregation, sort of a clumping, a congestion within the cytoplasm, and ultimately leading to cell death through a number of other mechanisms. If this particular virus interferes with that axonal transport, cell transport, nucleoporin transport, it can subsequently accelerate TDP-43 induced cellular aggregation. So it may be that if individuals had a TDP-43 mutation, or if they're sporadic who are undergoing some TDP-43 downstream effects, a viral infection like this could propagate, could help promote that mechanism. We're certainly not saying that this is a cause of anything. We're just seeing an association, and in this case, we're trying to understand more how that association is playing a role. More importantly, if it is, what we can do to help mitigate that.
Thank you so much. This next question is from someone who said that they are a caregiver for a pal who grew up in Guam from the age of 2 until 15 years, and that ALS doesn't run in his family. So your first slide mentioned pneumonium ALS. Did they find chloroviruses in this type of ALS, and how did they determine that diagnosis?
Well, and that's a little bit of a separate story. A good question, actually. The Guamanian form of ALS had a traditional theory behind it. And the theory was that after World War II, when corn substitute or corn substance weren't available after the war ravaged the area, they turned to another seed, something called the cycad cercinalis, and they used that to make a corn flour sort of material to make foodstuffs. Now, in this particular cycas ursinella seed, which is like a corn seed in a way, they could grind it down and use it for foodstuffs. It also contained this BMAA, and this is with early data. Some of that's been sort of questioned since then. But what we did see is after World War II, a very high incidence of ALS on the island of Guam within a group of individuals called the Chamorro Indian population. And what was really unique is these individuals also demonstrated evidence of Parkinson's and Alzheimer's within the same individual. I had a patient in my fellowship who actually was from this area, and it was just astounding that we would see all three neurodegenerative disorders within one individual, suggesting a common potential toxic contributor to three different neurodegenerative pathways. Um, Much has been done on Guam since then. This particular seed is no longer consumed, certainly, but there are still some other concerns what might be triggering it. We see pockets of disease in other areas around the world as well. We see pockets of MS in certain islands around the world. So there's much to be said about whether it's the genetics or the environment or both playing a role in certain areas. But the Guamanian form of ALS has always been intriguing, and there has been a lot of work done looking at a potential toxic exposure triggering the disease in a lot of the individuals who had that form of disorder. There may also be some genes we have yet to identify.
Thank you. Next question, I guess, is how do EMFs around large high voltage lines potentially affect people as well?
Honest answer, I don't have a good answer for that. I know that there's been an association of these high voltage lines with a number of different neurologic disorders. There's been some association with those lines with ALS populations, but I don't think any of them are clean enough to say it's that particular high voltage alone that's the trigger for any disease mechanism. Too many other variables are at stake. I can just say there has been some data out there in the literature to suggest that living near high voltage lines does have some health risks.
Thank you.
Thank you. Next question is, there are several immunologic trials that are launching. Which seem to be most interesting to you?
Well, the, so we're engaged. We have about 4 or 5 ongoing ALS clinical trials that we're participating in. They're all interesting. I just have to be honest. I think what catches my interest most is when we can attack multiple mechanisms at once. So the ones that are working upstream in the disease process, I think will have the most benefit. I'm Having done this now for nearly 35 years, there's a pessimism that can set in after a while with a single drug trial. I did my medical training, as mentioned, in Houston, and we were fighting a horrible disease at that time in the mid 80s, HIV, and we couldn't find one particular medication would be effective. It was that combination therapy, it was that triple approach with protease inhibitors that finally was able to take this virus into a dormant phase. Ultimately, that would be my hope for this disease. Can we turn it into a chronic illness rather than a devastating progressive disorder? And I think we just have to continue to add to our armamentarium. We just don't have a lot of good drugs we can offer our patients, let's face it. And the trials to date have been, well, they just haven't reached the endpoints we had hoped for. A lot of different potential reasons. I think it's probably because we need to have multiple targets being addressed. And when we only go after one target, it makes it challenging to see that global effect.
Thank you. So have they discovered things that deactivate or kill the chloroviruses at scale in order for us to kind of keep this under control?
Well, there's the in vitro and in vivo approaches. In other words, things within the body. No, we don't really have a means of controlling this particular virus. We haven't identified anything, certainly, mainly because we've never really identified it as being a problem in humans. So this is all relatively recent information. In terms of the in vitro, there's many ways that you can denature a virus through any bleaches or alcohol or a number of different disinfectants. So that alone, just similar to what we see with COVID, that alone isn't so challenging. The main issue, I suppose, would be if we had large bodies of water with large chloroviral infections, could there be a way to treat those lakes, those waterways, to try to reduce the population of these green algal blooms and subsequently, hopefully, see a reduction in the chloroviral population as well?
Thank
This
you.
is kind of similar. You may not have an answer to this, but there was a question of could phages be of use in targeting chloroviruses?
Well, potentially. I mean, we see a number of bacteriophage work being done with other diseases. At this stage, um, we don't have any work looking into that. We've also looked at the potential for even monoclonal antibodies against the viral capsid protein. Can we somehow go after a particular target on the virus that could inactivate it without damaging the cell itself? Apart from this sort of macrophage viral mechanism with the lectin receptors we discussed. So there's a number of different ways we're looking at it. But none of that has really been pursued. We've got an R01 grant underway right now to try to get some funding to do just that, actually, in a bit more detail.
Um, in what about Lyme disease or other tick-borne illnesses? Are those also under study for the same reasons or with the same approaches?
Well, I guess I can't comment on Lyme disease studies per se. It's not an area that we've been involved in. Um, Now, there's always the concern of infectious organisms playing a role. There has not been a lot of identified studies that definitively show Lyme's causing ALS. A number of different studies are out there. I don't think the overwhelming evidence supports that that's a major risk factor with Lyme's. There certainly may be some cases, I don't doubt, but with all the Lyme's exposures, if that in fact were a cause, one would expect to see quite a bit more disease as a consequence of that. And it may be like a lot of disorders that exposures can occur at a certain time frame in one's life, but yet the disease doesn't manifest itself till later on. Post-polio syndrome, for example, is a good example. Here's a virus that affects the motor neuron. They recover, and then 25, 30 years later, individuals start to become weaker again. Is it a burnout of the surviving motor neurons? Or could it be that the polio virus has remained dormant in the system for 30 years and becomes reactivated? So there's a lot we don't understand in terms of could some of the diseases we're seeing have been a consequence of an exposure to a toxin, whatever toxin it is, maybe decades before. So this is not to get too out in the woods here, I guess, in my answers, but there aren't any black and white answers to a lot of these issues, and I'm a big believer in you gather data, you do more experiments, you learn from what you have gathered so far, and you keep pushing it forward, but you do it quickly. This is a disease we don't want to wait on. That's my approach. We want to get answers as quickly as we can.
Thank you.
And that's where I just want to call out something that Dr. Ostro posted. He said that we have 15,000 serum samples from Guam. And basically that, happy to provide the samples to you if you're interested in testing them for chloroviruses.
So I think that's an excellent point that Dr. Ostro, I saw his note there as well. There's been the assumption that the BMAA could potentially be the reason, but that's been questioned, and I think that would be fascinating to look at some of those samples to see if any IgG1 to this particular virus exists. I don't know much about the terrain of Guam since Guam is waterborne by ocean. The virus we're looking at is freshwater aquatic, so I suppose it's certainly possible, and it would be very interesting to look at. So that may be something else we'll put on our list of to-dos here in the near future.
Gary, just to comment on that, we found some more records. There's a whole bunch of fresh water sources on Guam, and in fact was the water sources for the villages. And they didn't know what the infectious agent was or whether there was one, but somebody hand drew all these maps and figured out which villages were upwind or downwind of these fresh water sources.
Well, that's
And they
fascinating.
tested for some viruses they knew about at the time, but I don't think chloroviruses was one of them. But
Yeah. Yeah.
yeah.
Well, in that
By the
case,
way, I'm Lyle. You know Dr. Ostro. I mean, you know him personally, but I'm Lyle, right? So he's my dad, you know.
well, we know your dad and we know you, Lyle. So
Right,
yeah,
exactly. But yeah,
thanks for tuning in.
yeah.
Yeah.
We've got,
Question.
we'll
I'm
take a
sorry.
couple of questions. Go ahead, Anusha.
All right, great. So the next question was, could you speak further to any possible tie between NEK1 and the chlorovirus?
Uh, I'm not aware of any association between NEK1 and the chlorovirus. And I think we haven't really identified a genetic association yet at all to this particular virus. It's been sort of the other way around. We've looked to see, at least with the transgenic animals, where they have a genetic mutation, does the virus seem to propagate the onset of that phenotypic presentation from that mutation? And that's kind of what our data is suggesting so far. And that TBK1, for example that I used a bit earlier, showing that the COVID infection seemed to trigger the onset earlier, maybe by 2 decades from what we would normally expect. So I think much of our thought, based on the data we've accrued so far, is could these viruses be one of the tipping points for individuals that might have a genetic form of disease. That's something we've yet to prove, of course. Just our animal studies suggest that. But that's always been a question we've had. Why is it if individuals have a particular gene for a particular disorder, does it manifest at a certain decade in their life cycle? What is the trigger for that manifestation? And I think there's a lot to be said about toxic exposures, potentially even infectious agents triggering that onset process.
Thank you.
That may be true with other diseases, like, as I mentioned earlier, Epstein-Barr and MS. There's definitely an association there.
Thank you. You've we definitely covered from that end quite a bit in terms of where where might be the cause of ALS when it's related to CVs. But which viruses are closest to the structure of CV when we think about the actual structure of the virus and why it's so effective or why it's so well implicated? What is it structurally about it, and what other viruses are structurally similar to it?
There are a number of what we call large virus categories that we've looked at. As I've mentioned, there are 4 different classes. There's 43 different types. But this is a very unique virus. It has so many features in terms of viruses encoding their own SOD1. Rare, very rare in the virus world that you'll see this kind of a presentation. And even within the 43 isolates of a chlorovirus sort of genus, we'll see just a few that encode their own SOD1. We pick on that because that seems to be a potential common thread with the motor neuron disease process. I don't know of other viruses like this one that we could look at and compare. I'd probably have to defer that to a virologist, which I'm not. Okay, but that is something that up to now we haven't, in any of the other viruses, for example, identified any motor neuron-like human diseases. And then, of course, it could be that some of these viruses just haven't been looked at.
All right, so our last question for you tonight is, how can we help you in your research? Is there anything that we as a community can do, or other clinicians or researchers? What can we do to kind of keep this moving forward?
Well, no, that's a wonderful question, and I think you guys are doing so much already. I'll be honest with you. I think just getting the word out. To me, the most important thing in my research and in caring for my patients is seeing my patients as early as I possibly can, meaning it's unfortunate that it may take up to 9 to 12 months for a patient with this disease to be diagnosed. This is a very sad statement. And I think that it would be wise to sort of spread the word in terms of educating providers. We try to educate primary care teams on what to look for. Some of the features I mentioned in some of the earlier slides, from motor atrophy to fasciculations to other progressive processes. Seeing patients as early as possible to get them started on treatment options as early as possible. to me, is the most important thing we can do. Word of mouth, I think encouraging patients to get engaged in clinical trials is really critical. I'm a big trialist, I guess, by nature, and I love it that patients they donate their time, certainly, and that's a huge effort. Very difficult sometimes in rural settings where we live to actually get patients to participate because it's very arduous. It's very challenging. Just like your discussions before our talk on just getting to a vacation can be challenging. Well, getting in to see a physician or a clinical research coordinator once every month or two can be very challenging. Anything we can do to continue to make that happen and help out in that means, I think, is critical. This is how we're going to continue to push this field forward by having more and more data, more and more clinical research, good trials that are coming out. I'm fairly confident that we're making headway, we're making breakthroughs, never as fast as we'd hope, but you guys have a wonderful organization, and I think it's that whole concept of a relationship that means so much.
Yeah. There's a closing comment from one of our friends in the community that it's so challenging, as you mentioned. Usually trials want folks that have recently been diagnosed. What are the chances of someone being allowed in a trial with 5 to 6 years of onset? People want so badly to speak and to be able to regain the functions that they've had.
So what can be done by a community such as what you guys represent is to speak out to the trial drug companies. There's the EAP, the Expanded Access Program, individuals who don't qualify. And you're right, it's been ridiculous, in my opinion, how they've fine-tuned some of these studies to such a degree that only a cherry-picked few can enroll. And I've made these comments more recently. One of the trials we were involved in changed a lot of their parameters. Some of our comments may have helped to do that. We need to look at all patients, in my opinion. I think this is ridiculous that we restrict who can come into a trial. If a drug's going to work, we should be seeing it work in everybody. Now, there can be different biomarkers we use, but I think being vocal as a community towards the ALS trial community in particular, and saying if you have a study that you feel has to follow a certain parameter, then make a separate part of that study available for we call it early access, or everybody gets it. People that don't qualify for the study can get on the drug and see if it has any effect. I mean, follow them along. More importantly, see if it has any slowing in the progression. Now, I will temper that with 2 studies we've been involved in over the years where these were drugs that were used. They were easy for patients to get a hold of because their primary doctors could write these prescriptions for them. Minocycline was one of them several years ago. It was very difficult to fulfill this study. And what we found at the completion is that the individuals receiving minocycline did worse than placebo. And that was sobering. But if we didn't have a study to show that, people would have been taking this drug thinking, my goodness, this is going to slow down my disease, when in fact, it was making them worse. And so we need to have clinical trials out there. They've got to be almost always placebo controlled for X amount of time for the first several months, after which they need to open it up to the community to see if in fact it may have a benefit. I'd like to see more of that, obviously, but that can certainly be arranged. But drug companies have to work that into their trial budget before they start.
We would, and the kind of work we do here to allow people into digital health technology studies of all sorts. It's not just for observational insights, which of course we're doing to be able to help kind of untangle the need for greater stratification and understanding of the disease and how it affects different people, but also to prove the point to industry. And I think they're coming along that we can do a lot more remote monitoring within trials, and we can actually get a lot more detailed story of the different types of ways ALS affects different people with the different modalities that we can introduce, whether it's for speech tracking through our listeners' effort, or through gait and balance sensors, or fine motor sensors, or even respiratory sensors, all respiratory data capture, all from home. We're really passionate that these trials can and should be in the future redesigned to be from the home first, or as much as possible, to really expand the reach of people's ability to participate. So thank you for sharing our passion for that, Dr. Pattee, and thank you for tonight's presentation as well.
Thank you. Thank you for allowing me to share our results. And if you have any questions, feel free to forward them on. I'd be happy to try to answer them if I can. And these were wonderful questions you've asked tonight, I must admit.
That's one of our closing questions always is what's your take? How's it been to join the Everything ALS community for this conversation? And hopefully the questions were inspiring to you as well, and that you got a lot out of being with us.
Well, it's an honor to be able to do this, honestly. This is a disease many of us feel very passionate about. And for that reason, we're going to keep working, doing all we can to chip away at it. And, optimistic that we're going to find a magic bullet that'll make an impact in this disorder. But it takes the collaboration of many, many people to get that achieved. So you guys, this is wonderful what you're able to do.
Thank you again.